eukaryotic cells. So you need to know the differences and similarities. So make sure you are aware of the difference if they've got nucleus or no. So this could be animal cell, plant cell, or bacteria cell, and you can use it to explain the eukaryotic or prokaryotic cells. You then also have the specialized cells, so you might want to be able to explain the sperm that has a tail to swim. You might want to talk about red blood cell doesn't have nucleus because it's got a large surface area, and why they are specialized in their different ways, and so on. The other topic is linked to the cells, is also knowing the function of the organelles. So the organelles we're looking at is nucleus, ribosomes, and cytoplasm. So make sure you are aware of their functions, and don't say nucleus is the brain of the cell. Their job is to make sure everything is controlled. The next big topic we're looking at for the triple paper, a separate science paper, is the enzymes. For enzymes, you got the lock and key mechanisms that you need to be aware of. You got your active site, and you've got the substrate. And also we are looking, in terms of enzymes, we're looking at what denatures enzymes. So it could be the pH, change in temperature, denaturing or making it stop working if it's too hot or too cold. It could be the concentration of the substrate. So it could be any of these that we are looking at affecting the role. There is a required practical here linked to the enzyme about amylase. How does it work? And it hasn't been in the exam last year, so maybe look at that as well in detail, because it hasn't been. Again, linked to enzyme, it could be your digestive enzymes. So you need to know about like linked to the practicals amylase, but also you need to know about your lipase, your carbohydrates and protease. The lipase is to break down lipids, carbohydrates are for breaking down carbohydrates, and protease to breaking down protein. So these are all linked to the enzyme activity. The next topic I'm looking at is communicable and non-communicable diseases. So you can look at malaria, what causes it, and how to prevent. Some mosquito nets, taking tablets and things, and the protease that causes it. We're also looking at any other diseases. It could be plant diseases. Again, the color of the plant, if they're lacking certain minerals, what will happen to them in terms of colors or shapes? How will they change? So quite an important topic there. We could also link to diseases that are linked to virus, bacteria, or the fungal diseases. Cancer is another big topic in the exam. The factors what cause cancer, look at a lot of data linked to cancer. They can do data analysis there. If you're doing non-communicable diseases, again, if you're doing non-communicable diseases, it mainly is data and graphs that you look at the analysis of. Then we're going to move on to gene therapy. Now, gene therapy is of OCR and EdXL. So we're going to do gene therapy. Like I said, this is for only OCR and EdXL. So if you're doing those exam boards, so you got cystic fibrosis, you got other organs that can be affected, so they can ask you how to prevent it and what are the effects. Gene therapy, you can look at chromosomes sequencing, amino acid sequencing, what happens if you got additional or less chromosomes. Again, OCR and EdXL only. You're going to need the Punnett squares to do predictions of male or female, or anybody who's got disease or don't have a disease. So be careful there. Just make sure you draw the letters, capitals or lowercase. If it's capitals, then it's a dominant. If it's lowercase, it's recessive. When you're doing the crosses, and dominant's going to overtake the recessive genes. it's quite important that you're very clear on that one. So again, like I said, this particular one is linked into the OCR and Excel, but not AQA. AQA will be later on for paper 2, not paper 1. So please be careful that you're not getting the papers mixed up here. Again, in terms of gene therapy, you can be asked for amino acid sequencing, translation and transcription. You could be doing mitosis and meiosis. Again, like I said, this is for people who are doing OCR and Excel, not AQA. So the other topic that is OCR and Excel and not AQA is the food testing. So remember, this is just for these OCR and Excel students, not AQA. So in food testing, you got the root testing that goes from blue to green if it's weak, or purple or red if it's strong. You got the iodine testing for starch that goes blue, black. So you also got the Benedict solution. So Benedict's is basically, if you got anything, the glucose, that will change color from blue to, it could go green, it could go yellow, it can go orange or red, any of those shades. So you go heat up. There is a required practice link to that. We also, the major big topics for biology, regardless of how many times it's been in the exam, is photosynthesis. Always comes up. Really, really key topic. There is also a required practical link to this one. Right, remember the equation, a simple equation for yourselves, the practical, the limiting factors. So you need to know your limiting factors, you need to know your simple equation, and you need to know the required practical where you count the bubbles as you move the lamp to different distances. The one that hasn't been recently is respiration. Again, you need to know your aerobic and anaerobic respiration. You need to know how this affects when you're exercising. You normally get given graphs, comparisons that you need to do. The other final thing I'll say that might come up because it's linked to your required practical is the aseptic technique. So when you did had a petri dish on an agar jelly, you dipped your discs in different antiseptic techniques is a practical that they can ask you to explain.